Okay, hello students. Now I hope it is been cleared that what is rDNA technology from my previous lecture. So if you're very clear regarding the rDNA technology, then only you can proceed through this video. Okay, so we have seen that in rDNA technology we have used certain tools or say has the certain substance to complete the rDNA technology. So in today's lecture we are going to learn what are the tools that are required for the rDNA technology. Okay, I hope that rDNA technology, if I am saying the shortcut, then you are very clear regarding that rDNA technology stands for recombinant DNA technology. So let's start. That is 11.2, that is tools for rDNA technology. So what are the tools required for the rDNA technology? They are the restriction enzyme, polymerase enzyme, ligase, vectors, host organism. So in today's lecture, we are going to learn each of them in detail, step by step. So let's start. So first of all, let's see regarding the history of restriction enzyme. So in the year 1963, the two enzymes responsible for restricting the growth of bacteriophage in E. coli were isolated. So I hope you understood why I have written E. coli in italic, right? So if we are writing any of the scientific names, then it's a rule that the first, that is the genus name, should be in the capital letter. The first letter of the genus name should be in capital, as well as the rest, the species name should be in the small letters. Okay, and the next rule was that it should be, if it is handwritten, then it should be underlined, or if it is typed, then it should be in italic. That's why. So from in 1963, the two enzymes which are responsible for restricting the growth of bacteriophage were isolated from E. coli. After the two enzymes, the first one was adding methyl group to the DNA. Out of this two enzyme which was isolated, the one was adding the methyl group to the DNA, whereas the second one was cutting the DNA. I hope if you have gone through my lectures, then it has been very clear that which enzymes are responsible for cutting the DNA. Think, which enzymes are responsible for cutting the DNA? That is the restriction enzyme. As I said, molecular says the restriction enzyme are responsible for cutting the DNA. So the second enzyme which they found was actually the restriction enzyme. So restriction enzyme is being divided into two parts, that is the exonuclease, or say as there are two types of the restriction enzyme, that are exonuclease as well as the endonuclease. Now what is the difference, or say as the main difference between the exonuclease restriction enzyme as well as the endonuclease restriction enzyme? So the exonuclease restriction enzyme, what it would do? Exonuclease restriction enzyme will remove the nucleotide from the end of the DNA. It means that if this is a sequence, or if it is a strand of the DNA, it would cut from the end like this. Okay, and what would the restriction endonuclease would do? It would cut the DNA as a specific portion. It means that it would be having a specific sequence. It would recognize a specific sequence. Say for example, here if ATGCC something is present over here, and this is the sequence which the specific restriction endonuclease is targeting, then it would cut only that much specific portion. It would cut that much specific portion only, right? So this is the main difference between the exonuclease and endonuclease. See, this is the DNA strand which I have shown. Now this is the linear form of it. So endonuclease would cut from here. I'm sorry. Exonuclease would cut from here, that is from end, whereas the endonuclease would select a specific sequence. Say for example, this is the sequence. If I would say, it would cut this much portion only. Okay, I hope you have understood the main difference between the exonuclease and endonuclease. It's very easy, that exonuclease would cut the nucleotide from the end, whereas the endonuclease would recognize a specific sequence, and then after that it would cut the portion, right? Okay, the first restriction endonuclease, this is the continued portion only. The first restriction endonuclease which was discovered was hint 2. Okay, now this hint 2 always have a specific recognition sequence. as I said, it is an endonuclease, so it might be it is always going to have, not might be, it is always going to have a specific recognizing sequence. This sequence is having a 6 base pair. Okay, I hope you understood what is a base pair. Okay, so that 6 base pair, 6 base pair means is, for example, if here A, then here T, here G, then here C, like this. This is 1 base pair, this is 2 base pair, this is 3, likewise 6 base pair it is going to recognize. Okay, that 6 base pair is known as recognition sequence. Okay, there are 900 restriction enzymes that have been isolated from over 230 strains of bacteria. Now, how any of the restriction endonuclease is being named? So we are going to see the nomenclature, or say as how it is being named. Here I have taken an example, that is EcoR1. EcoR1 is an restriction endonuclease. Now, how it is being named? The first letter is been taken from the genus, that is E. coli, you know, right? So it is taken from the genus, that is E. Second 2, that is taken from the species name, means second and third letter taken from the species name, that is coli. R stands for either rough or smooth strand. Here it is a rough strand of E. coli, that's why here we have written it as R. If it is smooth strand, we would written as S. And last one, that is 1, that is a Roman letter. Okay, Roman number indicates the order in which the enzymes were isolated from the strain of bacteria. So there might be many strains of E. coli from which it might be given a name. So from that, the number 1 is being taken. So that's why we have taken we have named it as EcoR1. Now, each restriction enzyme would be having a recognition sequence, as I said. So recognition sequence would be always palindromic, would have palindromic nucleotide. Now, what does the word palindrome means? Say for example, palindrome means a word which is read as same from both the directions. Say for example, Malayalam, MALAYALAM. Okay, from left to right. If we read it from right to left, then MALAYALAM, same, right? So such type of things are called as, or such type of the word are called as palindrome. The second example is IEYE. I hope you understand what, I hope now you understood what is palindrome. So similarly, the recognition sequence of each of the endonuclease would be having a palindrome sequence. Say for example, this I am giving the examples of EcoR1. See, GAATTC, GAATTC. Okay, so this is the recognition sequence which is having how many base pair? 1, 2, 3, 4, 5, 6. This is also 6 base pair. So I hope you understood each of the points. Okay, now let's actually see what happening in this, or how what is the action of the restriction enzyme over here. So as I said, we have taken the EcoR1 example, that is EcoR1 restriction enzyme. This is your vector DNA. Vector DNA means vector, that is vehicle, that is plasmid, and this is the foreign DNA, means where your gene of interest been present. So in vector DNA we have got the specific sequence, means EcoR1's sequence. Similarly, in foreign DNA we also got the specific sequence. Now EcoR1 would cut like this, from here and from here in the vector DNA, whereas in foreign DNA it would similarly cut from here and here. Okay, after cutting, these are the strand we have got. So these are called as sticky ends. Why sticky ends? Because it is having overhanging. See, this are the overhangings. This is having overhanging. So actually in the cutting ends, there are two types of the ends, that is sticky end or as well as the blunt end. Here I am giving you the example of sticky ends where we are getting the overhanging, right? So this is an example of sticky end. After the cutting, that is after restriction enzyme, what is our procedure? To stick it with what? With the DNA ligase. So this is the recombinant DNA which you got, right? So see, you can understand from this figure very clearly that this is the vector DNA, that is our plasmid. This is the foreign DNA, okay, that is our we can say as alien DNA or a gene of interest where it is present. After that, we have cut it with specific ECO-R1 enzyme, that is disease and restriction endonuclease enzyme ECO-R1, which would recognize a specific sequence from both, that is from vector DNA as well as from foreign DNA. After that, it would cut it. So here we have got the sticky ends and we have joined it with the help of DNA ligase. So this is the recombinant DNA which you got. After this, what you will do? What is the step? So after this, it will be transformed into a specific host organism where it would be given the time period to replicate. So in this PPT, you have seen that this is the sticky ends. Overhanging ends are called as sticky ends. So as I mentioned, there is a second end, that is blunt end would be there. Okay, so note it down that sticky end is also called as cohesive ends, COHESIVE, cohesive ends. Okay, so specific restriction enzyme would create a sticky ends and specific restriction enzyme would create a blunt end. So the example of the restriction enzyme would create a blunt end is SMA1. So see, this is the recognition sequence having the again 6 base pairs. See, 1 2 3 4 5 6, having 6 base pair, but this SMA1 enzyme would cut and would create a blunt end. See, this is a blunt end. Here there is no overhanging, right? Exactly 3 here, nothing space between them. 3 nothing space between them. If you have gone through the previous PPT, then you can see that there are space between them, means sticky ends, overhanging. Here there is no overhanging. And the second one, that is blunt, and it's also called as flush ends. Remember this and note it down. Okay, so now whatever we have seen, this is the whole summary of that. This is the foreign DNA, this is the vector, that is plasmid. We are cutting both of them with the help of a restriction enzyme. Okay, we have cut this, we have cut this. After that, we would join it with the help of the DNA ligase, and this one is called as the recombinant DNA. After the formation of the recombinant DNA, we would transform it into a specific host. So this is the host, which is E. coli. E. coli would multiply, or say has, and okay, yeah, E. coli will multiply, and thus our gene of interest, or say as our recombinant DNA, would transform from the parent to the offsprings. So this is the end of the lecture.